I've been studying the population genetic of different kelps from the last 15 years, and what we found for giant kelp is that in the Southern California bight, the genetic diversity is the highest that you find around across the whole distribution of these species. So if you think about it, if you're starting a breeding program, Southern California bight is a pretty interesting area. You have all this variation that you can source from to start your breeding program. So it's really an ideal situation.
I'm a marine ecologist by training, and I've spent my entire career studying the ecology of kelp. Our Arpa-E funded research on the genetics and selective breeding of giant kelp applies cutting-edge techniques developed for land-based agriculture to a marine crop, which is very novel. I'm really excited about the potential applications of our results for domesticating giant kelp and improving its yield and material properties for use in biofuels and other products.
So the kelp that most people know is the big mackerel that is actually what we call a sporophyte. So this organism reproduces by releasing spores, and these spores pretty fast turn into male and female gametophytes. So these are haploids, and if you think about it, a kelp farm, what people often do to seed their farms is to go in nature, collect from a big mackerel, a sporophyte. They collect this part of the organism called a sorite that releases the spores, and they release them in their labs, in their nurseries, complete sexual reproduction, and you have the baby sporophytes, and you're ready to put things back into the farm, and then the organism grows and you harvest. What we're doing in our breeding program is a little bit different. So we do the same. We go out and we initially collect the spores and we let them develop into gametophytes. But then gametophyte can be controlled for not going to sexual reproduction and actually can reproduce them asexually. So you can produce clones of the same gametophyte. So these are haploid copies of the same identical genotype. So you have both the males and the females. So say if you can understand the genetic properties of a particular haploid genotype from male and from female, and that's what we do in the breeding program. We try to select for genotypes that are interesting. This means that we can cross male and female and get a superior kelp. So because we can grow the haploids asexually, pretty much unlimited, we can have as many copies of that particular sporophyte. This idea is really at the basis of our program.
To make difference in future of ocean-produced biofuels, not only we need to domesticate kelp, much more important we need to train people who will lead future kelp domestication and production of biofuels and food from the ocean. And I think that it is much more important for this project to speak not with me, but to hear from students who are actually doing it and will become leaders of the future.
Maintaining a gametophyte collection is the way to save and use the genetic diversity of giant kelp for different purposes. We have more than 4,000 gametophytes isolated for genetic region and sex. They are ready to be used in research, restoration programs, and kelp farming.
Our team is interested in the haploid microscopic gametophyte stage of the kelp life cycle for a couple of reasons. First, this is the point at which sexual reproduction occurs, meaning that we can see the emergence of novel traits such as increased resistance to climate change and increased biomass, two traits that are both of interest from an aquaculture perspective. But there are a lot of things that we don't know about the gametophyte stage. Specifically, we don't know if gametophyte fitness translates into sporophyte fitness. We're studying this by identifying the genes that are used for regular vegetative and reproductive purposes in the gametophyte stage. Our hope is that by identifying these genes and exploring the differences in translation of these genes across strains, we can identify those that have the genetic profile for increased fitness, and that this will ultimately translate into increased sporophyte fitness. And therefore, this will ultimately lead to increased yield and increased desirable traits for our crop.
One of the most important parts of a breeding project is a high-quality genome. We have assembled 3 separate genomes from different individuals from different populations in order to encompass as much natural variation as we can in our genomic resources. We have also measured the genetic variation of 500 individuals from 3 separate populations in Southern California.
And genome-wide association study helps us to actually get to that genetic loci that really changes the plant biomass, for example. So in our data, it's really nice because in 2019, we had a farm set up with about 500 individuals, and we actually have an idea of what genomic positions actually increase this trait in the kelp. Now, what has happened as a result is that we are able to make these genomic selection models which help us actually breed for these genotypes that have these genomic loci that can actually help us get kelp which are bigger and have higher plant biomass. And hence, for the future of kelp breeding, it's really important to actually make use of GWAS and genome selection models to make the future giant kelp varieties.